next week we're talking about synapsis and meiosis. Doing recombination as our topic next week. And so we have to talk very briefly now about synapsis and meiosis. Sorry, you're not looking at what I'm looking at. Next week. So we've got to talk about synapsis and meiosis briefly, just an introduction, remind you what that's about. The concept of crossovers, and two types of interference. And again, this is being recorded, so I'll just upload this to YouTube if you don't want to take notes at the moment. Two types of interference. Has anybody heard about interference before? You remember that? Probably talked about it many years ago in a genetics class at some point, maybe. So remember what meiosis is. Production of gametes, haploid gametes on this end, starting at a single diploid cell. Where does synapsis happen? What is synapsis? You know what where else do you hear about synapses?
In the neural system.
What is a synapse? That's where two cells
two neurons create a junction.
Come very close to each other.
Yeah, dendrites and axons.
So synapse and synapsis are related in the fact that it's things coming together. So this is synapsis. So there's replication. Each of your two copies, the copy of the chromosome you got from your mom and the copy you got from your dad, replicate. Those are sister chromatids. And then the pairs of sister chromatids synapse. That's this stage, metaphase 1. Okay, so that's synapsis. We're talking next week's paper about synapsis. This is what synapsis looks like in a cartoon form. Those two sister chromatids, the copy of chromosome you got from your mom and the version that just replicated, the copy, are the maternal in this case. So you get two different, each of these loops is meant to represent one double helix of DNA. So those two sister chromatids are very tightly intertwined, and they're attached to a protein complex, the axial core. And then the two copies, the sister chromatids from your dad, do the same thing. They're closely intertwined with each other, and they're also connected to an axial core. The synapsis comes from the zippering up using these proteins of those two axial cores. So you make a protein bridge between one dyad, two sister chromatids, and the other one. That's what holds all four of those things together in a tetrad of meiosis 1, metaphase 1. The point of this is to note that there are proteins involved in synapsis, the bringing together of the pairs of sister chromatids. This is what it looks like in real life. So these dark things are the two axial cores, and emanating are tons and tons and tons and tons of loops of those intertwined sister chromatids. This is early meiosis, before the chromosomes are really condensed. They're mostly condensed here, but what you're looking at in black there are those proteins, proteinaceous structures that run the entire length of the synapsed chromosomes. So there are 4 different sets shown here. So proteins are essential for synapsis. Okay. The proteins that are involved in initiating synapsis. It's a horrible title. I don't know why I wrote it like that. There's synapsis initiation complexes. Everything is something initiation complex. Replication initiation complex, etc. These are synapsis initiation complexes. A set of proteins that exist where those sister chromatids and pairs of sister chromatids are first brought in close opposition to each other. And that's where that zippering up process starts. So a core complex of proteins binds the pairs of sister chromatids together, and then that protein that cross-links the axial cores comes in and just goes along the chromosomes and brings the rest of them into synapsis. The protein in yeast that does the zippering, this transverse filament, is called ZIP1. That's one of the proteins that will be in the paper next week. The synapsis initiation complex has a bunch of different proteins in it. In yeast, it includes ZIP2 and ZIP3. So one of the problems with yeast is all the proteins and genes are named after their function, and so you get ZIP1, ZIP2, ZIP3, ZIP4, ZIP20 probably. They're all different proteins, they do different things, they just have very similar names. So the ZIP2 and ZIP3 proteins are found in these 6 synaptonemal initiation complexes. And ZIP1 goes the whole length of the synapsed chromosomes. Okay, so here is fluorescence of ZIP1. You can't see the chromosomes right now, but this fluorescence is in between 2 pairs of dyads. You get dyad fluorescence in between the synapsis, the synaptonemal complex, all these proteins, ZIP1, and then the other pair on the other side. You just can't see the DNA because it's not stained. So this is that, all those proteins that go across from the 2 axial cores, all the way down the chromosome. So there's chromosome all along one side of that, what is that, red? Whatever color this is. Down one side of that is one pair of sister chromatids, and along the other side is the other pair.
Is there 1 sic per synapse?
Ah,
Sorry.
fabulous. Good timing. So per chromosome, there are lots of synapsis initiation complexes. So in this figure, for example, you see 2. There could be lots. So synapsis initiates along all of the chromosomes, much like yeast arses. There are lots of positions where replication starts. There are lots of places along each chromosome where these what are we calling them? Synapsis initiation complexes form. So they can form at multiple places, and then you get the zippering up in between them. So the concept just simply is there can be multiple locations of synaptonemal initiation complex. Okay. Why is this important? So there's synapsis, the bringing together of the chromosomes. That's one cellular event. Then there's genetic crossover, which is why meiosis 1 is really important. Dredge it back up from your memories if it's been a while, but this is where strands of the different sister chromatids, once they've synapsed, break and exchange DNA segments. You recombine, you make new combinations of alleles on chromosomes that didn't exist before. And you pass those on to your kids. That's one of the main purposes of meiosis. Produce gametes, and then synapsis, in this case, produces genetic variation. I'm just reminding you. That's not entirely critical for the paper. But this is what that looks like. We replace the cartoon with an actual micrograph. You can see this is 4 sister chromatids, some of which are crossing over, exchanging genetic material between them. So you can see a chiasma, or the crossing, there, and there. So this set of sister chromatids is exchanging 3 different times. Aside from creating genetic variation, what else does this do? The physical process of recombination, crossing over, does something really important too. It holds together the tetrad temporarily during meiosis. Talked about, okay, so interference, last thing. Good. Is it random where those chiasma, the sites of crossing over, occur? Do they happen all along the chromosome? As we discussed briefly recently in Caenorhabditis nematodes, they happen very rarely in the middles of chromosomes, more frequently on the ends. That's more of an exception rather than a rule. In humans, recombination happens all over the place on chromosomes. There are some places that happens more frequently than others, recombinational hotspots. So
it varies between species to species.
So it varies from species to species, or genus to genus, or family to family, or something like that. But one way that crossovers are not random is that when you have a crossover happen, the likelihood that there's another crossover very nearby is much lower than the likelihood of having another crossover farther away. So this is more frequent than you would see 2 crossovers very close to each other on the chromosome. This is called interference. This crossover is interfering or pushing other crossovers far away from it on the chromosome. Don't know how. That's the big question for next week's paper. We still don't really well understand how interference at the molecular level works. We've observed that this is much less frequent than this. So we've got evidence that this happens, but how exactly is the information that there's a crossover happening here passed on to the rest of the chromosome? How is it that the chromosome knows how to spread out the crossover events? Now, that's genetic interference, because these are genetic events, the actual recombination of different molecules of DNA. Then there's what the authors of this next week's paper are going to call cytological interference. The synaptonemal initiation complexes also exhibit interference. That is, if you look along a chromosome, this might start looking familiar, if there are protein complexes where we can actually observe the locations of the SICs, maybe by looking for proteins like ZIP2, can we observe interference among these proteins? That is, if we see one here, is it more likely that the next one is far away on the chromosome than close? There's no known yet. There is no known link between cytological interference, whether these are spread far apart, and whether or not these have anything to do with the locations of crossovers. So as far as we know now, this and this are totally different. Where the chromosomes start synapsing doesn't necessarily have anything to do with where the chromosomes cross over. They could be two totally separate things. Because remember, this has to do with bringing the chromosomes together, just where that process starts along the chromosomes. This has to do with chromosomes breaking and exchanging genetic material. They could be two totally separate things, or they might be intimately related. Don't know. So this paper that we're doing next week aims, in essence, to find out. So this is exactly what they're going to do next week. I've described it briefly here on the board. Here's the approach. This top part is ZIP2 fluorescence. So they've used an antibody, I think, to ZIP2 proteins. So they can see that's the outline of a chromosome. The bottom here is ZIP1. So that gives us the length of that chromosome. Right? This is the fluorescence of the protein core that goes all along the synapsed chromosomes. So that gives us length. We've got ZIP2 fluorescence. So presumably these blobs in the top picture, how many blobs do we have? Like 1, 2, 3. Can't tell if that looks like it might be two next to each other. So we've got the total length of the chromosome measured by ZIP1 fluorescence. And we've got the relative locations of blobs of synapsis initiation complexes on the top. Do those exhibit interference? Are they evenly distributed? Do we find some cases where, so what they've done here, I've just cartooned it. They divide each chromosome into 10 different equally wide segments, and then they just catalog in what segments are the different SICs, the ZIP2 blobs. And so this is the main type of figure you're going to see next week, where we're looking at those 10 different intervals of equal size, 1, 2, 3, 4, up to 10. And then the percent of total foci, that is the fraction of the total blobs on a chromosome that are found in each of those tenths of the chromosome. So on average, 9% of ZIP2 containing synapsis initiation complexes are in the first tenth, 14% are in the second tenth of the chromosome, and so on. That's what you're going to be looking at next week, a lot. And we're going to find out, do these proteins exhibit interference? Are they evenly distributed along the chromosome? This makes it look like they kind of are. You're almost equally likely to see one of these protein complexes in any part of the chromosome. Do they exhibit interference, and do they have anything to do with where recombination happens?